In previous lectures, I talked about the null hypothesis, the p-value, and then applications using Fisher's test, chi-square test, g-test, t-test, ANOVA, non-parametric tests, statistical power, multiple test corrections. There's a lot of knowledge about p-values. Now, I'm going to tell you it's all wrong. Have a look at this example. Imagine we have 1,000 tests, for example, COVID tests, where the probability of a patient being infected or probability of the effect being real is 10%. That means that we have 100 tests where the effect is real and 900 tests where there is no effect. We set a statistical test with power of 80% and significance level of 5%. It's a good test. Because power is 80%, then we have 80% of all these things with real effect, we detect them. Power is the ability to detect the effect when it's real. Out of 100 real tests, we detect 80 true positives. The rest is 20 false negatives. Significance level means that 5% of all these tests with no effect will be detected as false positives. We have 45 false positives. The false positive rate is the ratio of false positives to all the tests where there is no effect, which is 45 divided by 900. This is 5%. We have 5% of false positives because we set the significance level at 5%. However, the false discovery rate, as you know, is different. It's the number of false positives, which is 45, divided by the total number of discoveries, the tests which we call positive, which is 80 plus 45. And the false discovery rate is 0.36. It is 36%. I know what you are going to say now. Well, we just need to apply a multiple test correction, for example, Benjamini-Hochberg correction, which controls false discovery rate. It's all fine when you have 1,000 tests, but all this tree can be rewritten in terms of probabilities. It's just easier to explain with actual numbers, but you can have only one test where the probability of the effect being real is 10%, and you will come to the same conclusion that in one test, the false discovery rate can be 36%. So if you publish a p-value less than 5% result, you have a 36% chance of making a fool of yourself. Okay, something smells fishy. What's wrong with these p-values?
Let's do a little test. I will read you a statement about p-values, give you some time to think about it, and then tell you whether your answer was correct or not. Let's play the p-values quiz. The first statement. A p-value of 5% implies that the probability of the null hypothesis being true is 5%. Think about it. No, it's a false statement. Null hypothesis is fixed. It's not a random variable, so we cannot assign it a probability. It's either false or true. That's it. The second statement. A p-value of 0.001 implies much more significant result than a p-value of 0.01. You have time to think about it. You want to call a friend? It's a false statement. In classical statistic inference, we set a limit alpha, let's say 5%, and then decide if something is significant or not. Also, statistical significance has nothing to do with biological relevance, so it's dangerous to say more significant. And finally, the last statement. The p-value is the likelihood that the findings are due to chance. You want to ask the audience? No. This is an incomplete sentence. P-value given H0 is true is the probability of finding this or more extreme result by chance. By skipping given H0, we completely change the sense of this sentence.
P-value. Given that H0 is true, it is the probability of observed or more extreme data. You know this definition already. It is not the probability that H0 is true. Or an alternative version, p-value is the degree to which the data are embarrassed by the null hypothesis. When we say that we assume the null hypothesis and all other assumptions to be true, and then from this we do statistical inference, all these other assumptions can be a little bit more complicated. First of all, you assume that all assumptions about the biology are correct, that there are no other effects, that instruments are calibrated properly, that experimental protocols are followed, that data are collected correctly, and there are no silly mistakes. If any of these things fails, if, for example, there are some additional effects you are not aware of, you can get a p-value less than alpha, and then you will reject the null hypothesis. But it's not because the null hypothesis is not true. It's because some other effect can affect your result. This is our big number one. P-values test not only the null hypothesis, but everything else in the experiment. Let's go back to the first example. Why do we get such a large false discovery rate? Well, we can do another Gedankenexperiment, a simulated population of mice. And there are actually two subpopulations. The first one, which contains 97% of all cases, is the null distribution. The mean is 20 grams and the standard deviation is 5 grams. We assume 20 as the baseline here. In the second, much smaller subpopulation, there is a real effect. The mean body mass is 30 grams. We are going to do a one-sample t-test against the null hypothesis that the mean is 20 grams. First, we need to find the correct sample size, so we do very quick power analysis. We want to detect the effect size of 2. This is Cohen's d, 2, which is the difference between the means, 30 minus 20, divided by the standard deviation, which is 5. We want a power of 90%, which is a pretty good, powerful experiment. We set significance level to 5%. And if you do pwr.t.test function in R with all these arguments, you will get a required sample size of about 5. So to get a powerful test, we have a sample size of 5. And now we draw lots of samples of size 5 from this population, from this mixed population. And then we plot the distribution of p-values. Let's have a look at this range between 0 and 5%. All results here are positive. We call them positive because p-values are less than alpha. But because we did a thought experiment, we know which of them come from the population with no effect, which is the brown shaded area, and which came from the population with effect, which is a green shaded area. These are true positives. They come from the real effect. The rest are false positives. If you calculate the false discovery rate here, it's about 63% in this case. 63% of our results are false positives. So the chance of making a fool of yourself can be much larger than your alpha of 5%. Why is that so? Have a look at a different example. This time, the no effect and real effect are equally prevalent in your data. It's 50-50. If you repeat the same simulation, you will get a distribution of p-values looking like that, where false positives are down at the bottom. You could hardly see them. And if you calculate the false discovery rate, it's about 5%. When the effect is rare, false discovery rate is high. This is the problem with tests where the effect you are trying to find is rare. Be aware of this. You can get a very high false discovery rate. The next question we might ask ourselves, what does p-value of 5% really mean? Let's assume that you get a p-value which is very close to 5%, 4.5% or so. If you do the simulation with 50-50% effect where false discovery rate is well under control and plot the range near p-value of 5 and select just one bin here, you can see these are true positives, these are false positives. This is our p-value, which is very close to 5%. And again, the ratio of false positives to the total positives is about 20%. So when you have a p-value which is close to 5%, the false discovery rate can be quite high. In this paper, Berger and Serker demonstrated using Bayesian approach that when you have a p-value of 5%, the false discovery rate can be very high. It can be more than 30%. To control this, you really need to use much smaller alpha. The so-called 3 sigma approach, 3 sigma in the normal distribution where p-value limit would be 0.003, only then you will get false discovery reduced to about 4%. When you get a p-value around 5%, the false discovery rate is high. The next problem with p-values. Let's do another Gedankenexperiment. We have just one normal population. There is 100% real effect, and the effect size is 1. This is Cohen's d. Then we do a one-sample t-test against our null hypothesis. If you have a sample of size 3, this is the distribution of p-values we are going to get. As you can see, it's all over the place. Yes, it's more likely to get a smaller p-value than a large p-value, but still you are going to get a lot of p-values which are 0.5, 6, 8, and so on. You can say that p-values are not reliable because in repeated experiments, you are going to get very different p-values. They are not reproducible. However, if you take a sample of size 10, and you see that your simulated p-values are concentrated close to 0, this experiment is much more reproducible. And the reason for this is that sample size of 3 will give you a power of only 18%, while the sample size of 10 will give you a power of 80%. Underpowered studies lead to unreliable p-values. There is another problem with underpowered studies. Imagine a population like this. This is a population where effect size is 5 grams. It's 5 grams from the null hypothesis. And we select samples of size 3 from this population. And we calculate p-values in a one sample test against 20 grams. All p-values which are below 5% are called significant. And the distribution of these significant samples is shown here. Now, the problem is, if you have a test like this with a very small sample size, and this test obviously is underpowered, and if your test brings a significant result, your effect size is skewed towards higher values. It's inflated. This is because these small p-values do a selection among all possible p-values, among all possible effect sizes, and the selection is toward larger effect size. So, underpowered studies lead to unreliable p-values, but underpowered studies lead to overestimated effect size, which leads us to a conclusion: when your experiment is underpowered, you are screwed. And this is not only a theoretical consideration, this is quite real and dangerous. In 2013, Button et al. Conducted a test. They looked at 246 papers published in neuroscience and then did some filtering and finally used 48 of these papers where they could find what the power of the test was in the paper. And this is the distribution of powers in various published papers in neuroscience. You can see that only a small fraction, about 14% of these papers, have statistical power more than 90%. Most of them are seriously underpowered. There is a lot of tests, a lot of papers where there is absolutely no statistical power. This is a real thing. This is a real danger. It happens over time. The next problem with p-values is the effect size. You have seen a figure like this, and I will tell you not to make it because it hides all the data. If you plot it properly, you can see that you have, this is actually real data from the real experiment we did a while ago. There were two fluorescent dots on DNA, and we measured the distance between them. And this is the distribution of this distance from many, many images. And there are two different strains, and one strain has a distribution which is slightly higher. This is the mean of the distribution. If you do a t-test, it will give you a very small p-value. And if you make plot like this, it will suggest that the second strain has higher distance than the first strain, and the effect is statistically significant. The problem is you have lots of data here, and the test, statistical test, is oversensitive. It's going to pick up very, very tiny differences. And when you plot all data, you will see that there is really no difference. There is total overlap between these two strains. And the difference is significant only because you have lots of data. Significance on its own, it's not really that important. Effect size is more important. You really need to look at the whole data to see what is important and what is not. When you have lots of replicates, p-values are useless. And also, significance does not imply biological relevance. There are more problems with p-values, unfortunately. Multiple test corrections. You had a whole lecture about this. You know how to do this. When you have 10,000 genes, you perform 10,000 tests. You use a Benjamini-Hochberg correction method, and you get a result, which is all nice. But in real life, experiments are complex, and they result in complex multidimensional data. And analyzing this data is not a straightforward task. You might first check whether there are any batch effects in your data, and you find, well, no. But is this a test or is it not? Should I apply multiple test corrections after this or not? And then you do analysis of your data. You search for one thing, you find nothing. Search for another thing, you find nothing. Then search more and find a result, and then there is again nothing. You did lots of searches, lots of tests, lots of analysis. Do you include this in your multiple test corrections or not? There is no obvious answer to it. This is a really tricky problem. It is not always obvious how to correct p-values. Let us summarize this. What is wrong with p-values? Well, p-values not only test not only the targeted null hypothesis, but everything else in the experiment. The chance of making a fool of yourself is much larger than alpha of 5%. When you get a p-value close to 5%, the false discovery rate is high. When the effect is rare, false discovery rate is high. When your experiment is underpowered, you are screwed completely. Statistical significance does not imply biological relevance. When you have lots of replicates, p-values are useless. And multiple test corrections are tricky. So what's wrong with p-values? Well, a lot, because statistics, unfortunately. This led to multiple papers published about it, p-values misunderstood and misused, and why most published research findings are false. Well, so obviously we have a problem. Let's pause for a second. Maybe Maybe it's not p-values that are wrong. Maybe there is something wrong with us. Have a look at this. This is a quote from a paper. There is some evidence that research which yields non-significant results is not published. Such research, being unknown to other investigators, might be repeated independently until eventually, by chance, a significant result occurs. The possibility thus arises that the literature consists in substantial part of false conclusions. When do you think this was published? Who made this comment, this warning? Well, it was published really long time ago in 1959 by Theodore Sterling. This warning had been made long time ago and been made again and again and again, and yet it doesn't help. People do not publish negative results. And the fact that people don't publish negative results can have dire consequences. This is an interesting analysis done on how false facts can be canonized in literature. Imagine that somebody did an experiment and published this experiment and published a result. This is only one paper published and people don't trust it very well. So imagine that this vertical axis gives you well, it's a posterior probability. It's a likelihood that the published result is correct. Initially, we assign a very small likelihood to this because we don't trust this result very much. Then there is another paper and someone published confirming experiments confirming that this result is true. So the likelihood of this being a proper fact goes up. Then someone published a negative result. That means that our likelihood drops again. And then again, there are papers with positive and negative results. And eventually we reach the probability level after which we decide this is canonized as a true, real fact. If it reaches a level below here at the bottom of this plot, we reject it entirely. The problem is, if people don't publish negative results, this thing never goes down. It only goes up until it reaches a level and it's canonized as a fact. This is a result from a simulation for tests with a significance limit of 5% and power of 80%. On the horizontal axis, we have negative publication rate, how much people publish negative results. And on this axis is the probability of canonizing false claim as a fact. When the negative publication rate is high, this probability is low. Eventually, it reaches 5% here. This is our alpha. This is supposed to defend us against false positives. However, if negative publication rate is low, if people do not publish negative results, just as in this figure here, this curve tends to go up until it crosses the limit, and we canonize this as a fact. And for low negative publication rates, this probability can be 60, 70, 80% easily. We can canonize false facts as real just because people do not publish negative results. So if you do not publish negative results, science is screwed. But there is a thin line between negative result and no result. You see, when you search until you find the result you are looking for, this leads to what is called data dredging or p-hacking. For example, you did your experiment and you found no result, so you might massage your data a little bit, maybe remove some bad replicas to see if your result improves. Or you can just ignore confounding effects. You did your experiment on 2 different days, but who cares? Sometimes you might not account for multiple experiments or tests. Or if your initial null hypothesis doesn't work, you formulate a post hoc hypothesis, a different one. You get a p-value of 6%. Well, we are almost there. Let's try again. Let's try to improve our p-value. And also, as I mentioned, not reporting non-significant results. Even if you are conscious of all these potential issues, the biggest problem is the mindset of an experimentalist. I did this expensive and time-consuming experiment, and I need a result. I really need a result. And it is hard to shake this attitude off. It is hard to accept that all your effort was for nothing. And this leads some researchers to cherry-pick promising results, to data dredging, to p-hacking. And please don't get me wrong, this is not malicious. It is usually done with best intentions of publishing a result. I'm not talking here about just blatant cheating. This is reality of scientific research, and you have to be aware of this. There is an evidence of p-hacking. This figure shows you how p-hacking is widespread. These people had earlier collected p-values from many publications and look at their distribution. And there is an excess of published p-values with respect to a curve which we would expect if p-values were random and only positive p-values or positive significant p-values were reported. The funny fact about this paper is that to show that there is an excess here, the authors looked at counts in two bins of p-values close to 5% and postulated the null hypothesis that these two counts should be equal. They did a statistical test and found a p-value, proving that other p-values are wrong. It gets even worse. In this paper, the authors tried to reproduce 100 published experiments in psychological science. They, instead of just checking whether experiments were done correctly, whether the power was all right, they actually reproduced entire experiments. And you can see the results, the comparison between original studies and replications. This is the p-values. In original studies, all p-values are less than 5%. In replications, we are all over the place. And this is the effect size defined in the same way. Effect sizes are positive, are quite large here, while in replications, the effect size again is negative, positive, or zero. They managed to reproduce only 39% of published results. And the problem is deeper than you might think. It is not only about conscious or unconscious p-hacking. It's about the choice of the statistical method. This is an interesting paper. 29 teams involving 61 scientists used the same data set to address the same research question, whether football referees are more likely to give red card to dark-skinned players than to a light-skinned players. The data contained a lot of information about footballers in a few European leagues in years 2012 and 13, their country, club, age, height, body weight, skin color, and many other variables which might or might not be relevant to the study. The researchers were asked to give the odds ratio, its confidence interval, and the p-value to say whether the effect is statistically significant or not. This is the result. We have 29 studies. The point here is the estimate of the odds ratio. Odds ratio are 1 when the dark-skinned players are equally likely to receive a red card than the light-skinned players, twice as likely or 4 times more likely to receive the red card. The red dot represents a significant effect, the gray dot represents a non-significant effect. And there are several studies which agree with each other, they are very similar. Some of them give a very different result. Some of them are significant, others are not. Some of them have smaller error bars, others are large. These two studies gave a very large effect size, very large odds ratio, but they also suffer from large error bars. So there is a problem here. We have exactly the same data set and it's analyzed by experts, by statisticians. And they give you a short report how they analyze data. You can pick any of these reports at random, open it, read it and say, yep, this is a reasonable approach. This is a proper statistical approach. And the result, well, I think we can trust it. And yet they use different approaches, each of them correct. And they came up with different results. Oh dear. Well, p-values seem to be broken, or maybe we are broken. Well, we don't know. What do we do? What can we do about this? Well, first of all, before you do your experiment, and that's very important, before you do your experiment, talk to us, talk to data analysis group. We will help you designing a good experiment which will not suffer from all these problems I mentioned before. And now I'm going to show a slide with lots of stuff in it. I will quickly go through this, but it's the intention of this slide is for you to keep it and come back to it later. So before you do the experiment, specify the null hypothesis. You need to know what you are looking for. Design the experiment properly with randomization, so there are no batch effects, and make sure you have enough statistical power. You know how to do this now. Quality control is very important. Otherwise, some crap comes out in statistics later, and I have to deal with this. Now I'm going to say a few controversial things. First, I would suggest ditch the alpha limit. Use p-values as a continuous measure of data incompatibility with H0. This is not a standard approach. This is not what you can read in statistical textbooks, but alpha limit can be dangerous, and calling things either significant or not is a false dichotomy. P-value could be just a measure of data incompatibility with the null hypothesis. And remember, we assume the null hypothesis. You never ever say that large p-value supports the null hypothesis. If you get a p-value close to 5%, it only means worth a look. It's not a real result. Reporting a discovery based only on a p-value less than 5% is simply wrong. You need follow-up experiments. You need validation, independent validation. If you want to demonstrate your discovery using only p-values, you have to use much more strict rule, for example, 3 sigma rule. And finally, when you report your results, always report the effect size and its confidence limits, not only a p-value. Show your data, all data. Do not make dynamite plots. Do not use the word significant. It's a risky word, and the meaning of this can be confusing. And do not use asterisks to mark significant in your figures. The important thing, validation, follow up experiments to confirm your discoveries, and publish negative results. I know this is very hard, but you need to try. At least in your paper, when you search for various things, and a few of the things have p-values larger than 5%, you should still report it. We looked for this, we didn't find this. We looked for that, we didn't find that. Finally, American Statistical Association statement on statistical significance and p-values. I won't read it, but you can read all these points. I talked about most of them. If you follow this shortened link, you will find the paper. It's a paper worth reading. It's only 2 pages. It's very concise, and it goes through all these 6 points about p-values and their interpretation. I will finish this lecture and the entire series with one last positive thought from Clarice Weinberg, who is a biostatistician at National Institute of Health in the US. Propensity to misuse or misunderstand a tool should not necessarily lead us to prohibit its use. Thank you.